Here we have a picture of a spinal cord that's been stained for substance P. It's actually part of Nigel's research material. And we can see this nice staining in this region, which would be what part of the spinal cord?
Dorsal.
Dorsal horn, which is gray matter. All right? Down here, what's this structure right here? And then the ventral median fissure helps you orient yourself. All right? Here's a higher power toluidine blue stain thing. We don't see the whole spinal cord. We can still pick out the ventral median fissure. We can see the central canal, and the dark staining would be the ependymal cells that line that canal. We can see the dorsal horns, ventral horns, and these darker specks that are relatively large, even at this magnification, would be cell bodies of large motor neurons. All right? So white matter, white matter, white matter. So it's opposite what you see in the brain, where the gray matter is external. All right?
Are there any cell bodies in the dorsal horn?
Yes, there are. They're smaller neurons, and if you look closely and are well fixed, then you can pick them out. What's nice about the ventral horn neurons is they're large. They kind of hit you upside the head. Okay, this is a paraffin section of a nerve, and it's quite nice. I don't know, there may be 12 fascicles here, right? Individual fascicles that are held together by what kind of loose connective tissue? We call it epineurin. So this is all epineurin all around here. What defines and delimits an individual fascicle is a thin band of dense, irregular connective tissue that we call, all right? And then the endoneurium is kind of a concept thing, the loose connective tissue that surrounds each myelinated and unmyelinated fiber, all right? Now, if you osmocate the tissue, osmium fixes lipids. The myelin sheath of myelinated fibers is 80% lipid. You get a totally different look at the tissue. So here's an osmocated nerve fascicle. We don't see the whole thing. We can see perineurium right here. Right, these little black spots would be adipocytes. They're full of lipid. They turn black when you osmocate them. But here you see a nice little donut, right? A dark ring, which would represent the myelin sheath, surrounding the pale staining what? Okay. So keep in mind, what you see is dependent on how you fix it. That's why I took the time to talk about how you prepare these tissues on the very first lecture. Here's a higher power, and actually, this is crap. We're going to use our own research material in succeeding years. But you can see, myelinated sheath axon. This would be the perineurium that's somewhat separated artifactually. So the endoneurium would be this tissue in and around here. At this magnification, with this preparation, hard to see the unmyelinated fibers, okay? But they're there. If we look by electron microscopy, and I put the slide in, but it doesn't really matter, we see a little bit of perineurium here. We see this nucleated structure, which is the nucleus of the Schwann cell that's elaborating this myelin sheath, okay? Over here we can see another myelinated fiber. We don't see the nucleus in this profile, okay? These structures here represent unmyelinated fibers. Here we can see the nucleus of a Schwann cell, and we've got this unmyelinated fiber has 10 axons that I can see. So that's kind of a difference between a Schwann cell that invests a myelinated fiber, which only invests one domain of the myelinated fiber, one Schwann cell per one region. And again, if you've got a meter-long motor axon, you're probably, say, a millimeter per Schwann cell. Do the math. You can figure out how many Schwann cells that are covering, forming that myelin sheath. Here, you've got an individual remic cell, Schwann cells are sometimes called remic cells, that invests, I counted 10 there, so multiple. So here we've got 1, 2, 3, 4, 5, 6, 7 unmyelinated fibers, all right? You might see an image like this on your exam. So what is it illustrating? Myelinated fibers and unmyelinated fibers. Sarah?
The one in the middle on
This one right here?
Yeah, that's a Schwann cell or an
No, no, this is a peripheral nerve. You know, that, and again, I should have made that clear. I was following up from that peripheral nerve bundle. Oligos are found only in the brain and spinal cord white matter. All right? The myelinating fiber of the peripheral nervous system is the Schwann cell. But it doesn't always myelinate. Hence, you can get unmyelinated fibers. All right? Okay, high power, out of context shot of the muscularis propria. All right? It's so high power and out of context, I can't tell you whether this is intercircular or outer longitudinal or vice versa. But what we clearly have here is what? Yeah, myenteric plexus of Auerbach, if you want to spit out the whole thing. That's a beautiful neuron. You can see it's clearly a nucleus with a nucleolus, and you get this almost purplish cast to the pericardium or the cytoplasm. All right? Okay, that was the nervous system. What are we looking at here? Okay, and we know that because of the striations, and the nuclei are peripherally oriented. What about this nucleus right here? Excuse me? Actually, we're looking on top of the fiber. So that nucleus is peripheral, which is we're looking downward. Okay? You don't see centrally located nuclei in skeletal muscle unless there's some sort of pathology going on. Higher power shot. You can still see nicely the striations. We can see these peripheral nuclei. We can see this loose connective tissue that's got a capillary in it. So this connective tissue would be called endomysium or endomysium. Exactly. Here's that cross-section of this is actually a cryo-section that Nigel showed in his lecture. We can see that skeletal muscle fibers are polygonal in shape. And, you know, we're talking 100 microns or so here in diameter. And remember, they can be up to several meters long. We can see the peripherally oriented nuclei, and here we've probably got a capillary running in the endomysial space. This is from Diane Shelton's lab, another beautiful, this is a Gamori trichrome. I don't expect you to know that. But we can see clearly skeletal muscle fibers cutting cross-section, and then we have this encapsulated structure, which is a muscle spindle. Remember, that's the structure that helps regulate muscle length and the rate of change of the muscle. So you've got these modified muscle fibers. There are two types there, you don't have to know that. And two types of afferent or sensory structures that go to the dorsal horn, and you form a monosynaptic reflex arc with a motor neuron, right? So depending at, you know, it's the patellar, well, it's not quite patellar because that's a Golgi tendon thing, but it's the same sort of thing as a reflex, all right? And this actually, in a Gamori's trichrome, is a nerve. This is a histochemical stain for cholinesterase, all right? If you think about the neurotransmitter is acetylcholine, and you've got, once you've released it and it's bound to its postsynaptic receptor, you want to get rid of it. Otherwise, you've got to, if you inhibit cholinesterase, you have a nice insecticide. That's, a lot of insecticides are cholinesterase inhibitors. So basically, the organism just goes into tetany, and that's that. But this stain is for that enzyme then that breaks down the acetylcholine, and therefore it's a nice marker of motor end plates. So you can see that you've got motor end plates in all the regions where you've got that beautiful scarlet stain.